Hi, Ching. How are you? I hope I'm saying your name right. Um, the unmute button is all the way on the bottom right hand. There should be a little microphone symbol, and there you can unmute. Can you hear me? Um, hey, Kirko. Hey, Manas. Hey, Jared. How are you? Hi, Kirko.
What's up? How you doing today?
Good, good. Thank you. I have to figure out. Yi Ching, can you hear us?
Yeah, I can hear you.
Okay, then maybe let me ask in the chat. Okay. Can you unmute? If not, sometimes it helps to just come back to the room if the unmute button doesn't work. Or maybe switch from Wi-Fi to cellular data or the opposite. Sometimes the first time using the app, it's kind of buggy. We recently learned that. So if you want to come back, like restart the app, then hopefully it works then.
Good morning, friends. Does everyone have their audio functioning?
Hi, I'm not speaker. Can hear us or not.
Uh-huh.
She said she's still figuring it out, but I'm not sure if she can hear us
Or if
or
she
if
can.
she maybe can't unmute. But then it would maybe help.
Have you tried, do I pronounce your name Yishin? I guess you can't speak. Maybe leave and come back if you're not able to unmute. That's often helpful. But good morning, Katarina. Good morning, Kirko.
Hello?
Yay, we hear you.
Oh, nice. Yeah. Nice, nice. Yeah, I'm still trying to figure out how to use this, but since I can work, perfect.
Great. Yeah, sounds great. And we still have around 5 minutes, so we still have some time. How's your Saturday going? Everything good?
Yeah, it's perfect. Thank you so much for inviting us.
Yeah, no, thank you so much for coming. It's an honor having you. So you recently started working for Neuralink, or
Yes, I actually started 8 months ago. So I finished my defense on February, and I just joined the company right after I finished my defense. So yeah, it's on February. It's been a while.
okay,
Yeah,
nice. Should be
I'm still
exciting.
trying to. Oh, yeah, it's pretty exciting. It's a really good company, so much fun, and a lot going on in the company. And yeah, and I think we will have an event, a recruiting event actually, like end of the month. So yeah, if anyone in the clubhouse in the room chat is interested, super welcome to join. Um, yeah, I'm
Oh,
trying to
nice.
my colleague, Major is supposed to come. I don't think he joined the room yet, so let me just try to talk to him. Just one
Okay.
second. Okay, yeah, just got in touch. Okay, yeah, he's here. Nice.
Oh, hi. How are you? Ming Teng. I hope I'm saying your name right. And I hope you can hear us. And if you can, the unmute button is all the way on the bottom right hand. There should be. Oh, I think he maybe pressed the wrong button. Let's see.
Oh,
Okay. Should be back in any minute. In the meantime, feel free, everyone, to share the room with people you think that would be interested. And we will start in around 2 minutes. So, yeah, will be a really interesting and exciting room. Um, so, um, do you think he will manage by himself to come back, or let me see if I can find him here on Clubhouse to invite?
yeah, let me ask him quickly.
Oh, I already pinned him in. So let's see. If not, I can. Should I share the link again in the email? Maybe. Hi, Denise. How are you?
Hi everybody, doing great. Really excited for this talk.
Oh yeah. Also, I should share the paper link in the chat.
I just talked to Minjin. I think he will join in a second.
Sure. Yeah, we are not. It's weekend. Happy weekend, everyone.
Happy weekend.
There you are. How are you? So to unmute, it's all the way on the bottom right. There should be a little microphone symbol. Yep.
Hello?
Hey. Yeah,
Hey.
perfect. We can
Nice,
hear
we can
you.
hear you now. Yeah, right on time.
Yep. Right on time. We'll start in a minute. I hope everyone is doing well, having a great weekend. And yeah, I'm so happy that you both could make it. That's wonderful. Thank you.
Yeah, thank you so much for inviting us. I actually never used a clubhouse before, so it's a new experience for me.
Well, wonderful. I'm glad. You. I hope you'll enjoy it. Let's not be.
Yeah, since I hear a lot of talk in science society, so it's just nice that I can listen to other people's talk too. So it's a great platform.
Yeah, I saw you. Thank you for coming. I saw you in another room the other day. That
Yeah, yeah,
was
yeah.
nice.
It's so interesting.
Okay, I think we can slowly start. Probably people will keep coming in, but we'll start with introductions and so on, and then we'll go from there. So, welcome everyone to Science Society today, and of course, a special welcome to Yi Ching and Ming Seng. I hope I'm saying your names right. Please correct me if I'm wrong. And let me give the audience a short introduction about you, and we'll go from. And then usually Victoria asks a couple of interview questions, if that's okay with you. And then the stage is yours for presenting your really cool and interesting research. So, Dr. Yiqing Wu, she when she did this work, she was at the Department of Material Science and Engineering at the Curry Simpson Institute for Bioelectronics at Northwestern University, and that's also where she got her PhD. And she did her bachelor's degree at the Tsinghua University, and she did it in material science and engineering. And Dr. Ming-Tseng Wu, he did his PhD at Yevgenia Kuzorovsky, associate professor of neurobiology, and in the Department of Neurobiology, also at Northwestern University. And now he is a postdoc in Dr. John Rogers' lab at Northwestern University. Yeah, and congratulations for this amazing paper, and thank you for being here. And Victoria, the stage is yours for your questions. Thank you.
Thank you, Katarina. And so wonderful to hear about your work, Yijin and Mingjing. And please correct me, help me pronounce your names correctly. We
And the
are,
pronunciation is
yes,
perfect.
pardon, go ahead, sorry.
I'm just saying the pronunciation is perfect.
Okay, wonderful. Thank you. So, in addition to the work that you're about to present, it would be fantastic to learn a little bit of background about you both and learn about that background as well, because we're interested in science through people. Where Science Society is about bringing science to people and also exposing our guests as scientists to people as real people, so we can hear what interests you. And so my question is, if you can think back through your life about a time when maybe you first noticed that you had a particular interest in science, then we would love to hear about that. And that could be in your childhood or during schooling or from a parent or anything that just comes to mind, that's something that connected you.
I think I can start. This is a really good question. So for me, if now I look back into like my life when I was a child, I was really interested in detective stories. So I just hope like honestly, as a child, I just hoped I can be a detective and just solve crime problems and stuff like that. But it turned out that I'm a scientist now, so probably like different choices of my careers at this moment. But yeah, I think that will be the starting points where I feel like I'm just interested in figure out problems and trying to solve problems, and then I got into like math, chemistry, and physics when I was in middle school, and it's just so interesting that it's kind of like playing games, solving problems, and I don't know. I just feel like it's just so much fun to solve a problem and just figure out what's exactly going on. And science is it's all about like asking questions, answering questions, and find out a solution. And I think that's basically the starting um, where I feel like I'm so interested in science and that's yeah, if that's the question. But there's just like totally another story where I feel like I'm interested in more like medicine, medical devices, and for that it's more about my families, and my grandpa died early when I was in middle school, and um, because of cancer. So at the time, because of that, because I already interested in science, and I just have an urge to study more medical parts of the science. So in that case, I can help people. I can save people. So yeah, so that's basically why.
Yeah, thank you. Thank you so much for the extra detail. And it's really interesting to hear that you started out wanting to be a detective and then how you applied that curiosity towards science and through your tragically, I'm sorry that you lost your grandfather to cancer, and you've used that experience though to motivate you in medicine and to solve problems there. And so thank you for that answer. And Mingzhen, the mic is yours.
Yeah, I was actually a medical student at the beginning, so I'm not on a scientific trajectory. But I think like during my intern time, like in China, during my internship in the hospital, I saw there are a lot of patients suffer from like pain and like some emotional suffering. But the problem is that the therapeutics are like not sufficient to manage their pain and sorrow. So that motivated me to become a scientist instead of like being a physician. So I think after that I joined a lab whose focus is on neuropathic pain. So like during my undergraduate research, I like was focusing on the mechanism of neuropathic pain and the mechanism of morphine dependence. Morphine is most used therapeutics for neuropathic pain these days, but there are a lot of side effects, physical dependence, and potentially addictive to many patients. So I was studying that mechanism and trying to minimize these side effects and trying to develop more like therapeutics. That's what I did in my undergrad. Uh, so, and from that experience, I think I should pursue a PhD degree, potentially in the US. So I enrolled in Northwestern University and joined Janice Lab, where I start like I switched to the field of neurobiology and the mechanism of emotional suffering, like for example mood disorder, like depression and bipolar disorders, and I want to know the mechanism of them and I studied like really promising rapid antidepressant, which is ketamine, that it works really fast and like a single dose of ketamine can have the antidepressant effect for like a long period, like approximately 2 weeks. So that's really promising, but we do not know the mechanism of it. And my thesis research was focusing on ketamine's antidepressant effect, and we found that it's actually the dopaminergic modulation, like in the major prefrontal cortex, and the synaptic plasticity was involved in ketamine's antidepressant effect. And I think during my thesis research, I also started the collaboration with the Rogers Lab and with Yixin, and I realized these new technology are really promising to manage. Like they have like a lot of potential to manage these relatively chronic like mood disorders and other neuropsychiatric disorders. So I decided to like switch my discipline again, like from neurobiology to that entire engineering world, trying to find some solution to these like global health burden in the society. I think that's it.
Yeah, thank you, Mingzhen. And thank you for the detailed journey as well. It's the thought process when you're changing paths and determining direction. It's challenging to leave one and go toward another. And I'm it's very moving to hear how much you're motivated by the desire to address emotional suffering and as you said, to learn the mechanisms behind that. And it's a common thread. Both you and Yijin are talking about how compassion is a driver in your research toward finding answers to help people. So at this point, I would like to pass the mic back to both of you for the rest of the room, and you can go into your discussion, and then we, the moderators, are here to assist for when you have questions. Perhaps you'd like to have a Q&A following your discussion, then we can moderate that. And sometimes guests in the audience will put questions in the chat for you, and we can share that with you as well. So at this point, thank you so much again for being here, and the mic is yours.
Thank you so much. Yes, we're so honored to be here and share our recent paper that's published in NatureCom. And I think so. Basically, for the agenda here, I'll just talk a little bit more about the backgrounds of this paper and introduce technical parts of the paper, and then me and Jen will go into the animal experiments with this the systems that we built. So yeah, I think I can just get started with the background. I think everyone has the slides already. So if you can open the slides and we can get started with the first slides. Okay, yeah, hopefully everybody opened the slides already. So yes, so I'll start with the background. Um, so implantable devices have been used for neuromodulation to treat a variety of diseases such as depressions, Parkinson's, or chronic pain. And the desire to study implantable devices with multiple stimulation models had grown due to the early experiments, which showing that stimulus can be therapeutic. So for example, on the right, electrical stimulation, on the right of the slide, electrical stimulations have been shown to minimize the symptoms of Parkinson's disease, and recent studies have attempted to combine stimulation therapies with sensing to monitor the therapeutic process. So simultaneously, device considerations have moved towards low modulus materials, wireless operation, lightweight design, etc. So if we can move to next slides. Of the various combined stimulation therapies that I just mentioned, optical and pharmacological has exceptionally interesting property. This is because light is a fundamentally orthogonal physical stimulus to normal brain function. Therefore, if neurons or drug molecules decide to become light sensitive, the off-target effects is very small. So light sensitivity can be introduced using two different pathways. The first one is optogenetics, involves introducing a virus which forces the cells to express membrane protein, which are light sensitive. These group of membrane proteins, widely termed opsins, are then illuminated, which alter the ability of the neurons to experience an action potential, and this highly targeted chemical optical stimulation can therefore promote or regulate synapses in a group of neurons. And in contrast, optopharmacology involves a more simple concept of introducing compounds which is degraded upon light illumination, and it will release cargo materials that can alter membrane protein, which also alters the neural function. So general process for introducing light sensitivity to the brain is by injecting either a virus or a drug into the brain, connecting to a fiber optic cable for light stimulation, and then monitor animals' behavior as a result. Optogenetic stimulation has shown to be effective to minimize the symptoms of Parkinson's disease. Okay, so let's move to the next slide. With this in mind, unfortunately, the previous tools for optofluidic stimulations are somewhat lacking. Large syringe pumps and tubes or laser system and long optical fibers are typically necessary for drug delivery and for optical stimulation, respectively. This large and steep cannula and fiber, as well as the physical fetters, actually damage the brain tissue, as well as prevents observation of animals' natural behaviors. Recent advances in wireless optogenetics open up new opportunities for optogenetics. However, parallel optical and pharmacological modulation are still lacking. Okay, so if we move to the next slide, based on this logic, a wire-free optofluid device and materials must be carefully designed to solve these challenges. This also according to the materials paradigm, since I'm a materials engineer, and we decide which structure and processing steps necessary based on the final materials performance. The device basically consists of 4 components. Firstly, the fluidic package, which applies the pressure gradients to induce fluid flow in a consistent manner, must be small, lightweight, require low power, and brain compatible. The probe carries the drug delivery and light stimulation properties. Materials must be compliant, yet able to penetrate the brain while localizing the drug and optical stimulation. The power supply must remain lightweight while small and provide sufficient power. Ideally, the control system will allow the greatest degree of programmability possible. If we'll move to the next slides, beginning with the pump. Various pump strategies are possible, but the basic operation typically involves an actuator, which applies a force on the diaphragm, and the diaphragm, made from flexible materials, then deform, and therefore pressure gradients develops within the drug reservoir, inducing fluid flow towards the outlet. Various previous arts have been developed, such as peristaltic pump, piezoelectric pump, and thermally activated pump. But these pumps either heavy or bulky, or require high voltage, high temperature operation, which are not ideal for a miniature implantable system for virus or drug delivery. So then, if we can move to the next slides, to solve the previous challenge, I present this electrochemical pump. It consists of interdigitated electrodes, which can generate oxygen and hydrogen species based on electrolysis reaction, shown on the left top photos over here. So the virus or drug and electrolytes are firstly loaded in the drug reservoirs and pump chamber. After initiation of the pump, electrolysis reaction happens and the gas build up within the pump chamber applies a force on the flexible membrane, which pressurizes the drug reservoir on top to enable fluid flow through the microfluidic outlets. This device is capable of low power operation while maintaining a very low temperature change, with both chambers remaining below the body temperature of most mammals, which is favorable in delivering virus and temperature-sensitive drugs. So if you can move to next slide. After developing the pump, I built this multilateral optofluidic system, integrating the pump with this serpentine interconnect for spatial flexibility and easy implantation. And the multi-components neuroprobes consist of microfluid channels for drug delivery and micro-LED for optical stimulation. A microfluidic bulb is also built for flow modulation. The photos and details fabrications of microfluidics are actually included in the backup slides. The device is powered by near-field communication protocols, which take advantage of magnetic resonant coupling between the device antenna and the emitted radiation radiation cage from the RF module. With my colleague Abraham's help, we developed the circuitry and user interface for real-time programmable control over micropump and micro LEDs, and the wireless multilateral multi-model platform creates unique options for various modes of operation for neuromodulation. So from here, I'll hand over to Mingjun to talk about different operations of the device and how it applies to animal experiments.
I think before I go into details, I'll introduce a little bit motivation from the biology side. As you know, the optogenetics and pharmacology they're really useful in neuroscience research and they're really powerful. But the problem of the conventional approaches of optogenetics and in vivo pharmacology is that they're externally like tethered with fiber optics or like fluid deliver channels. They need to connect the amino like to some external power source or like drug delivery source, and it really affects the naturalistic behavior of those animals. For example, if you have multiple animals in the same cage and all of them are tethered with some like external cable, they will not like engage in social interaction with each other. So if you want to study social behaviors with optogenetics and these tools, it's gonna create a huge confounding factor to the interpretation of the results. So I think that's our initial motivation to make this technology wireless, so that we can like put a really small device on top of those animals, and they can like social interact with each other like normally. And even though if you have some like complex environmental setup, they can move in that setup like freely without any physical constraint from the externally tethered cable. So that's our motivation, but there are like some previous published study of the wireless platform. They're, I mean, they're they're very nice and wireless, but the I think the form of those previously published platform is still like this is still relatively large compared to the size of animal, especially for mouse. It's like putting a bowling ball on top of the animal's head. So we want to like uh, reduce the size of this platform. So we get rid of the battery and we use the RF to power this device. That way we can only use a small receiving coil, like for those devices, and give the get the power from the radio frequency from the antenna outside the animal's behaving cage. So that's some background. And in this case, we also upgrade the system by introducing the near field communication uh, system that we can update the parameter of the light stimulation and drug delivery in a real-time manner. That way, we will have a lot of combination of the operation to study animal behaviors when using these devices. For example, if you can see slide 7, so we can operate a single channel at each time. We can operate different LEDs targeting different brain regions, and we can operate different fluidic channels to deliver different types of drugs in multiple brain regions simultaneously or separately. So by doing that, we first we need to validate this technology is actually working in animal behaviors. So we came up with a like really simple behavioral paradigm that where we can activate the secondary motor cortex to generate the rotational behavior of the animal. So the way we did that is we preload some virus solution that encode channel rhodopsin in the wireless uh, optofluidic device, and we can deliver channel rhodopsin virus into the secondary motor cortex. And after a few weeks later, when channel rhodopsin expressing in these pyramidal neurons in motor cortex, we can give blue light stimulation to drive the rotational behavior. So that attempt was successful, and another reason we choose for rotational behavior is that we can reliably quantify the degree of the the rotational behavior and the speed and the amplitude, and it can be easily observed by visual inspection, I would say. So we will know we will know if the device is working as soon as we start the stimulation. And as mentioned before, a very important functionality introduced in this new platform is that we can control the parameters in a real-time manner. We can combine the operation of drug delivery with the optogenetic stimulation. So it means that it opens the possibility of a new field of research, which is the photopharmacology. That we can deliver some photoactivatable drug, or like, and then we can use light to change the conformation of that drug and to make the drug active to the neuronal group that we are targeting. So we can use the light to control the we can use light to control the function of that delivered drug, and thereby we can manipulate the activation of receptors on these neurons, uh, like in a high temporal, in a relatively high temporal resolution. So the first thing, so we want to try is we used the blue light uncaged neurotransmitter. So it's called ruby glutamate. So if it receives the uncaging light, single photon of blue light, it will release the glutamate, which is an important neurotransmitter from the caging group, and it will act on the neurons and modulate the activity of these neurons. And we also did some in vitro experiment to validate this approach is actually working. So I used some E-phase recording of a parameter neurons and applied the ruby glutamate to the bath. And after we gave the blue light stimulation, we can evoke some post-synaptic excitatory post-synaptic current that can be blocked by glutamate receptor, which means this compound is actually working. So then we can infuse the ruby glutamate in the secondary motor cortex, and we can use blue light from the device to uncage ruby glutamate to activate the neurons in the secondary motor cortex. And if we do that, we can generate a significant rotational behaviors. But without the light stimulation or without the ruby glutamate delivery, we won't generate any rotational behavior, which means that this approach is working. And a really promising thing of this technique is that we have a lot of different type of caged neurotransmitter. For example, we have caged dopamine, caged like neuromodulators, for example, caged oxytocin. So we can use this approach to study the function of those neurotransmitters and neuromodulators in a really precise way. And finally, another thing enabled by this platform is that we can study social behaviors. We can control up to 256 devices in a single field. Although we don't need that many animals to study social behaviors, but it's just demonstrate that we can do a lot of more things than the platform published in previous studies. And Yixin, do you like to summarize here, or I can go ahead and summarize?
Yes, yeah. Thank you so much, Yunzhen. So yeah, to summarize, we build a system, this wireless autofluorescent system that can allow real-time programmable control with various operation modes, and we also demonstrate in vivo of the capability of our platform by optogenetics, optopharmacology, and integrating optogenetics on free moving mice. So yeah, so this is our talk. Thank you so much. We're open to any questions.
Yeah, thank you so much, Yiqing and Mingzeng. This is really amazing. Also, especially that you used all that you are making all these different caged components. I keep telling people to do that, and you're the when I was in George Augustine's lab at Duke, we had the postdoc. She would make all kinds of caged components, but in other labs, that's really not used that much. But it's so useful, right, to have this temporal and localization. So it's really exciting. Do you have maybe first results with using caged oxytocin, caged dopamine, or are you what are you planning to use it for? I would imagine caged oxytocin would be really interesting to look in autism models, for example, because we know that there's like a difference there in oxytocin levels and receptors.
I think yeah, I can explain a little bit on that. So the reason why people develop all these caged neurotransmitter neuromodulator is that for conventional pharmacology, they just deliver dopamine or oxytocin into the brain regions, then and the action of these compounds, they're constantly on, but it's not the case for the real brain system. So basically in brain, these oxytocin and dopamine, they are released in a pulsed manner. So they're not like release a whole lot of oxytocin and dopamine into the region and then stop firing. They are try to release, so they're trying to release like in a really, I would say in a really precise way. So the reason why we develop these caged compounds is that we want to mimic the real uh, release pattern of oxytocin and dopamine and other neurotransmitters in the brain, so that we can use light to control the activation of these uh, receptors for the certain neuromodulators. And then something we can do is to study the function of these neuromodulators in some behavioral setting. As mentioned by Katrina, we can study the mechanism of like autism in to see if the oxytocinergic modulation is involved in autism. For example, we can deliver the caged oxytocin in the middle prefrontal cortex, and then try to mimic the release pattern like of these oxytocinergic neurons in the brain, and to see if we can modulate the autism-like behaviors in these animal models.
Yeah, thank you so much. It's really exciting. Does anyone have questions? Denise, Kirko, Joyce, Wisam, you joined the stage. Please go ahead if you want to ask a question. Okay, I'll. Is you guys can hear me, right? You guys can hear me well.
Yes, we can all hear you.
Okay.
Go for it, Katarina.
Okay.
We're just all thinking, there's a lot to think about, which is great. Go ahead, please.
So I really like the wireless optofluidic device components that you. So how deep can you go? Like what's the depth of the 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 delivery that you can go, and did you try out how well it stays in place in when if you need to reach deep brain regions? Thank you.
Yeah, that's a good question. So for our NeuroPro, it basically consists of micro LED, microfluidic channel, and this micro LED is built on top of a copper a layer of polyimides, copper polyimide, sandwich a thin film, and then we laser cut it into into a specific shape where we hope it to be for sure miniature enough that it will not damage, will have like a minimal damage to brain tissue. And then it can also, as a substrate, to hold on to the microLED and microfluidic channel. So as for your questions about how deep it can go, it can be customized into any length because we can just design the shape that we want and we laser profile it into a specific shape. So it can go into like more shallow brain region, like multicortex. It's shallow. It's like more shallow, yeah. And then deeper into the deep brain. So yes, it's totally customizable. Customizable. And then for microfluid channel, so microfluid channel basically it's a soft polymer based tube that we made where there's like a small channel within this soft polymer. So it can minimize, it has low modulus. It can minimize the mechanical mismatch between the device and also the brain tissue, the soft brain tissue. So in that case, with this microfluid channels, we can deliver drug with like minimal damage to the brain. So with this microfluid channels, it's made from photolithography methods to build up the channel shape, and then finally it's also defined by laser profiling. So that's why it can be made into a very small size where it can have a width of only 50 to 100 micron. So yeah, so that's why it's like very small. It's customizable. You can go into a deep brain.
Yeah, that's wonderful. That's really exciting. And I can. So I saw that you have an LED in the end. Did you also try out a string of LEDs that you can like program maybe in different patterns? Let's say you want to activate different depths or different brain regions along the way in different patterns. Would that be possible?
That's a very good question. So with this, it's also possible that we can make a deep enough or a long tube, a long probe with opening at alongside at different heights of the probe. So it's pretty straightforward to build, to design a microfluid system with different outlet locations. For example, as a separations of like 1 millimeter, separating all across different depths of the probe, and then we can realize this delivery, like a logic of delivery at different locations, and maybe we can program it into with like a different time lapse. So we can deliver the first one at a more shallow depth, and then another one with like a deeper depth. So in the case, we can realize um, this delivery of multi-location and multi-time points.
Yeah, that's so interesting. And do you also can you record at the same time, or maybe even have a mini camera at the same? Because if you could make an automated feedback mechanism if necessary at some point.
Exactly. Yeah, that's a very good idea. So there for sure that's something that we want to build like future in our system with like a feedback with micro camera, or further it's also possible to build a sensor right on top of this device. So now it only has stimulation, but it doesn't really have like a sensing platform for the feedback system. So that's actually a lot of efforts have been put into building sensors where we can integrate a system with stimulation and sensing at the same time, and it can definitely be a more powerful system for feedback control. This is something that's even like my lab and also a lot of other scientists in other lab are working on.
Yeah, I think that's something we are currently working on. We're currently working on the electrical sensing and fluorescence sensing. So with these sensing modalities, we can make the optogenetics in like we can make a closed loop optogenetics. If we detect a behavior event like from the camera, if we detect some specific neural signal that is related to a type of behaviors, then we can like use that closed loop system to manipulate neuronal activities in that pattern, and we can artificially build like connections between like different brain regions like through that closed loop system.
Yeah, since we mentioned dopamine previously, there also, I think a paper is coming out, probably like later in the year or next year. It's also a paper in the group which which is a dopamine sensor, that can be potentially be built in with our system that potentially we can uncage dopamine and then monitor the dopamine concentration, and in that case as a closed loop system to monitor the behavior of the mice.
Could I
Yeah,
ask a
yeah. I just wanted to say. Now I remember. I couldn't remember our guest speaker. We had the guest speaker from um, Professor Andrea Armani. She made this nano-sized molecular device capable of sensing and altering cells' bioelectric fields. I know she only tried it in like cultures, but maybe that would be a cool person to collaborate with. It was really impressive, her talk. I can share the paper if you guys are interested. Thank you. Go ahead, Wisam.
I just had a couple technical questions. I was looking at the slide at the bottom of your presentation with the implantation. It looks like you might be, making holes in the skull for where the probes are going to go down, and you were talking about flexibility and the depth you can insert these. I'm wondering about how much flexibility there is in terms of choosing which brain regions. In particular, I work in the auditory cortex, so I'm wondering about if it's flexible enough to try to approach the temporal lobe, auditory cortex, if nothing else, from directly above. And then just general questions about the surgery looks pretty straightforward, but I'm curious, how, the success rate of surgeries, the lifetime of the probe, and whether the device is reusable between mice, like having completed experiments in one mouse, can the devices be reused in other mice?
I think in terms of flexibility, we have two different types of design. So if you are targeting like the same brain region on two different hemispheres, we have this fork-like design that the implantation is very simple. We can just hold the device and and drill the hole on the bilateral side and try to insert the probe. And we have another design that for more flexibility, we have this flexible design. Basically, the two probe will be like separately connected to the device. So that way we can insert the like probe into different brain regions because they are not interconnected with each other. So you can try to target, for example, auditory cortex with one probe, and you can target maybe frontal lobe with one probe. It's just I think it's just the choice for the specific experimental setup. And we have also tested the like precision of the targeting with this flexible probe. We just compared it with like fixed design, and we found that the targeting precision is quite identical for these two designs. For like I think for experienced experimenter, the targeting location is the targeting precision is quite similar, but it like other type of surgery, you need to get some experience to really get it work. But the overall process is quite simple, and we have like protocol paper published, I think early this year to like for the surgical procedures, and I think it will be helpful for a starter like to like do surgery like this.
Yeah.
Uh,
sorry, can you give me a sense of what that precision is? And I can't tell from the image just how wide the probe is. Does it require a full blown craniotomy, or can you just do like a burr hole size kind of?
Yeah, we can do a blur hole. So
Okay,
the dimension of the probe is not. It's pretty similar to those fiber optics. I think
okay,
it's like 300 millimeter, 300 micron in thickness. So it's like yeah, that's pretty similar for like conventional fiber photometry or optogenetic fiber implantation.
okay. I'll be quiet after this, but can you give me a sense of what the targeting precision is?
So let me find find the number, the paper. So we compare it with the fork design. We just try to compare the precision at two different brain regions if you are targeting them at the same time or targeting them separately. I think it also depends on like the blur hole you drill, but I think the precision is within 100 micron. That's what I remember.
Okay, thank you.
Yeah, I believe all those details are included in the paper. Because I remember they're in the supplementary for the precision of the probe is in inserting. So
Awesome. Yeah,
so
I shared the paper with my lab. I'll definitely check it out. Thank you for answering my questions.
Yeah, I think for both of these techniques, it's approximately 100 within 100 micron like variation.
nice. Yeah. Yes, I also want to answer the question that you asked about the reusability of the device. So at this moment, because we're using cements and use cements to secure the device, and the cements can interfere with the soft microfluidic channel. So if we remove the whole device, then the microfluidic channels can get damaged. For sure, there's some improvements that we can do in terms of securing the device. So if that's the case, then potentially we can reuse the microfluidic channel, but in this case, we can only reuse the electronics parts of the device. But if we want to reuse the device, then we would need to remove microfluidic channels and replace the microfluidic channel, and also for sure need to clean up the whole device, the reservoirs, sterilize and all this stuff. So yeah, that's the answer about the reusability.
Thank you. Thank you.
I know you joined the stage, and Kyoko and Joyce, if you have questions, just flash your microphone, and then in the meantime, yeah, go ahead.
Yeah, hi, Katerina. Thank you very much. Great topic and research. Uh, I have two questions. And the the mice used here, do they change behavior compared to other beings in their surroundings while they're being tested on this subject? And what are the actual difference in between the fluid water uptake, intake, and the behaviors during this tests? Yeah, thank you.
I think we during the specific behaviors we're looking at, which is the rotational behaviors, uh, we don't see much behavioral response generated in those animals that did not express channel opsin, which means that the effect we're seeing is option dependent and activity dependent. But I think for the daily routine of the animal, we usually single house them after the surgery, so they recover pretty well from the surgery because for the body weight and fluidic intake, food intake, they're not significantly different from those wild type animals. I would say that after appropriate pain management after surgery, these animals are quite normal compared to the animals without the surgery.
Oh, thank you very much. Just a follow-up question, because we have a tendency to see that what we're being exposed to, if it's blue or red light, it will affect how we behave, and
Mm-hmm.
hence the water intake, for example. So yeah, I don't know, you anything you want to add?
Sure. So that's a problem of using blue light. So we're trying to switch to use red light instead, because for mice they don't see the light at relatively right wavelength. So that part, the red light won't affect animals' behavior, but blue light, if it's glowing inside the brain, I'm sure that they'll see that blue light flash. But it really depends on what specific type of behaviors you're studying. But I think the overall trend is to use like right shaded options to cook for behaviors, because there are like two advantages of that. The first, I mentioned that the animal don't see much of the red light, and second thing is that if the red light can actually penetrate like more depth inside the brain, it means that we can target a relatively larger group of neurons with red light. And I think on the device side, we can also increase the power of the red LED by a lot by introducing the capacitor bank, like to store some power, and we release it when we need them, so the red light can be really, really bright. So yeah, that's the directions we're moving, because there are like some concerns of using blue light in the field.
Very interesting. Thank you very much. Thank you. Yeah.
I also saw a lot of questions in the room chat, so I think we can also answer some questions over there. I think the first question is, have you evaluated the effects of RF field on mice, and is it used only when needed or always on? So for the effects of RF field, based on our study, we did not see any like negative effects of the RF field on mice. And since RF field is pretty commonly in our daily life too for human, we have our phone and a bunch of RF signals everywhere. So yeah, so this for sure, it's highly possible that there's a chronic effect, but since it's a short experiments with RF field only on when it's needed. Also, that's the second question that you asked. But yeah, so that we did not see any negative effects on the mice. So that's the answer. And then Minjun, do you have any answer or follow on this question?
I think I saw the question in the room chat, like how far away is the optogenetic from utilizing in human subject. I would say we still have a long distance to go, because the conventional way of like doing like optogenetic in animals, that we use virus to express opsins in these neurons, that's gonna create some problem for the translation, like to human research. But I would say there are a lot of studies that are going on for like using optogenetics in non-human primate animals, like monkeys. I think there are a lot of efforts of putting on to like to make optogenetic translate to like human studies. But I would say there are still a long way to go. But I think this wireless neurotechnology may offer some solution to that, because for human research, for human application, we cannot expect to put some cable to the human subjects. But for wireless, it's gonna be a lot better. But still, uh, it's not something that are ready to apply in human research.
Yeah, I also want to kind of also I saw the comments over here. A crisis might help to sell it to the public. Yeah, it's really funny. So I also want to follow up on this question about how far away is optogenetic from utilizing human subjects. As I remember, correct me if I'm wrong. I think there's already products using optogenetic on eye therapeutic. So I think that's definitely a step forward. But I think for FDA approval of devices like optogenetic devices used in human subjects, even electrical stimulation devices. It's been studied for like what 50, 100 years, and it's still if we're gonna use in human subject, then that's a lot of evaluations beforehand on not just mice, on NHP to figure out if it actually have like any kind of side effects on humans. So that's one of the reason why optogenetics is it's still a long way to go for optogenetics to be used on the human. And on the other hand, optogenetics um, it's it's using genetic modulation and introducing a gene into a neurons that's a very sensitive topic that it may potentially like raise up a lot of questions about is it safe, is it going to be like other kind of side effects, um, and is it going to be consistent, is it going to be effective after certain years, since cells um, can also have after probably 1 or 2 uses, the cells can have like response that has like a lesser response to the stimulation. So yeah, a lot of questions about that too. I guess yeah, I would say probably several years still, maybe even 10 years.
If I can add, there are clinical studies to restore eyesight with optogenetics in humans. They use blue and yellow, so yellow light to inhibit and blue light to activate. I haven't looked at them again, but they're ongoing for a few years now, maybe 3 years or so. I was in the first conference of applied optogenetics in Boston, and there they shared already that they were starting. But I haven't looked at it again. So yeah, if so, to restore eyesight, it's pretty. It's not deep in the brain, so it's pretty low risk. But to go deeper in the brain, it's probably a higher risk. But yeah, check those out if you're interested.
Hi, this is Joyce. This is all very fascinating, and I don't know if you've mentioned this yet, but and I understand it is a long time frame. But is there any condition you have in mind that you think, besides what Katarina just mentioned with the eye, that you think it's more likely to be used in sooner than perhaps other conditions? Anyway, thanks. I'm done.
I think for peripheral nervous system, it's gonna be sooner than the central nervous system. I would say like for managing neuropathic pain, like for example in the spinal cord, it's gonna certainly, because we have like more knowledge on the like spinal cord probably than the brain, and we can like try to manipulate a certain circuit for that like gate control mechanism of pain, and like try to suppress neuropathic pain like in the spinal cord, or like try to suppress the neuronal activity in the dorsal root ganglion, like this peripheral nervous system, like try to manage the neuronal activity acutely in that. basically, I think in the sensory system, in the motor system, it's gonna be faster than the, like, for example, emotional control or like things like that.
Thank
That's
you.
what
That
I'm thinking.
that's
That's what I'm thinking about.
yeah, that makes a lot of sense. So maybe it could be, 8 years for that versus 10 or 15 for the brain.
Yeah.
Yeah, there's also a question about what kind of lifetime does the probe have from Jared. So this is a good question because for microfluidic channel, we do have um, a lot of studies on how to reduce or there's a problem that the probe can get clogged. Because it's the reason is not really that clear to us if it's a scar tissue or if it's there's some tissue got pushed into the microfluidic probe during the surgery. So we suspect that the first one maybe like the first reason, and we change the location of the microfluidic probe outlets to the edge of this probe instead of right on the tip, and it seems that it worked pretty well in preventing any tissues directly going into the probe during the surgery. So that helps us in declogging the microfluidic channels, so extend the lifetime of the probe. And for scar tissue, on the other hand, that's something that we haven't figured out fully because for some mice, after the surgery, it seems like there's just less blood and less damage to the tissue, and then it's just lesser chance of clogging of the microfluidic channel. But on some mice, it may just be more severe, probably due to the surgery process, probably due to the mice itself. We're not sure about that, so that can affect the lifetime of the probe. But at this moment, if the surgery is going well and if for the initial 1 weeks after 1 or 2 weeks after recovery is working properly, then the lifetime of the probe that we have at this moment is up to 2 to 3 months. We didn't do any additional experiments more than 2 to 3 months, so not 100% sure what is the longest lifetime, but yeah, that's the lifetime of the probe.
I had a follow-up question on the probe. What is it composed of, the materials itself?
Yeah, so this probe is composed of 3-layer polyimide, copper polyimide for controlling and for giving electrical signal to micro LED. So that's the substrate, and then it's encapsulated in perlin. It's perlin C. And then on top there's it's a polymer based microfluidic channel built of PDMS. Yeah, so those are the materials used.
Yeah, thank you. And how far away for the wireless one? How far away can the probe be for you being able to trigger it? In case you have like more elaborate behavioral studies or overnight and so on. How close does the trigger have to be?
I think so for this version of device, we just trigger it like using like the so we can actually trigger the behavior from like very very far away, like just like using the computer. But the size of the experimental setup is a little bit limited for the reason of power transmission. I think we managed to uh, I think for 10 square feet, that's probably the largest environmental setup you can use for now. And so for this platform, the control system is actually through the near field communication. But for the next generation of devices we're working on, we can use Bluetooth like to control the devices, and so then it will depend on the distance for that Bluetooth communication system. So basically, it can be like several meters away, like 10 meters away. It's quite comparable with like a normal Bluetooth connection, like for example to headphone, like things like that, and we can actually control the device on our portable devices, for example cell phone, like from like relatively long distance. That won't affect the behavior of the animals.
Yeah, so for near field communication, it's more short distance, and for Bluetooth, it's more long distance. So for near field communication, normally it's actually built within the field, so the animals need to be moved. So animals can move within the RF field, and then that means that the field rate the sizes of the fields is very important. So at this moment, yeah, as Mingjin just mentioned, 0 to 10 meter will be the size for the NFC, and then for Bluetooth, on the other hand, it's more long range. It's 10 to 100 meter. But on the other hand, Bluetooth require higher power operation. So in that case, we need to add battery for the system, and it may increase the weights and the size of the device. So there's some trade-off over here depending on what kind of experiments we're aiming for. So yeah.
Yeah, that's really interesting because, for things like sleep patterns, where you maybe want to modulate different factors of the circuitry or social behaviors, I think that's really important to have these wireless devices, and where you can have some range if you want to be more, this automated systems. I don't know if you ever saw them. There are some companies that make this fully automized behavior systems where you kind of the mice get monitored 24 hours all the time during different all kinds of behaviors, and so you get like 24-hour data, which is really interesting. Yeah, Joyce, go ahead. Joyce, did you unmute on purpose, or you're just having a hot mic?
I'm
I think
sorry. I
Joyce.
guess I unmuted on accident. By
I have a follow-up question. So in the lab, is that a radio quiet zone? Are you trying to minimize interference with the wireless signals, or does that not matter?
Yeah, that actually matters. So we did have like a specific space, or it's more matters in terms of communications of the wireless the antenna to our system. So to laptop in this case, but there's like a connector in between the antenna and the PC. There's some like tuning channels, and that's actually affecting by other RF signals. So we do have like a specific space for the animal experiments.
Yeah, I think that matters a lot for sensing, but for stimulation, I think it doesn't matter that much.
Yeah, I think mostly it's just the showing of the cage that we're using for communication. That's but yeah, it's not really a big deal. We can just improve the system by like improve the current setup that's built by Neurolox. This is a company that Professor Rogers established and 3 years ago. So it's building kind of like a connectors that's in between the RF antenna to the laptop. So so in that case, we can control the system by writing our code in laptop, and then we can tune the RF signal by this RF tuning box that they develop, and in that case, we can apply the RF signal to the antenna, which is surrounded by the cage. But yeah.
So the cages are made out of plastic, probably?
Yeah, it's acrylic. Yeah, it's acrylic.
makes sense a metal would interfere to some extent with
Yeah,
all this radio signaling.
yeah.
Well, does anyone have more questions? Because if not, I would maybe get back to ask one more time about the caged. How precise can you be with the uncaging in that system? Like, what's the scale of precision? Like, not the temporal, more like how many micrometers you can be triggering the uncaging.
It also depends on the concentration of the solution, and then also since light illumination, we can change the power of the LED to change the light illumination. So in that case, for these kind of experiments, we can do a um, or like in vitro benchtop experiments in hydrogel to figure out the illumination level, and with that, and also the diffusions of the drug without cystine, and with this combinations of um, studies of diffusions of the drug in hydrogel and also the light illuminations within the hydrogels, we can determine the distance or the effective distance of the drug in the brain. So in that case, we can also tune the parameters of delivery, duty cycle, and the powers of LED, duty cycle LED. So in that case, we can tune to our target distance. But of course, like in vivo experiments, maybe totally different. Not totally different, sorry, maybe slightly different. But that will probably need like additional experiments. But there are just a bunch of studies on how to change a hydrogel into like a matrix, which is more similar to brain tissue. For us, we're using aerogel, but then it's also possible that we can um, kind of cross-linking the gel into like a matrix that's more similar to brain tissues with textures, different kind of textures, and we can potentially study a little bit more about its diffusion, drug diffusion, and illumination throughout the textured hydrogel. So that's pretty interesting subjects that people are working on too.
I think for the spatial resolution, it's not that high because that's a single photon caging experiment, not two photon caging. I would say it's gonna uncage the compound like yes, like 200 micron, like something like that.
Is there any potential for this to be used as treatment for folks with epilepsy?
Yes, I think there's there's a like paper that's currently under review about like treating epilepsy. So it's basically can introduce a closed loop system with the EEG sensing. If the sensing part detects like EEG signal that is like a seizure like, so it can deliver some drug like into the brain region that triggers the seizure to suppress the onset of the seizure. But it's still on the pharmacology side.
Got it. Thank you.
Jared in the chat asked, does scaling up from rodents mean you'll be able to add new features to the device?
Yeah, that's totally possible. By scaling up from rodents to, are you talking about like monkeys and NHP and human? So if that's the case, then potentially the device can be bigger, but on the other hand, the power required for um, for the functionalities for the simulations may be higher too. So that's something that we need to evaluate, figure out, and that will be different questions in the power budget and the size of the whole systems and what exactly kind of functionalities that we can add. So like new features, it's the question is what kind of new feature? Is it sensors? Is it other stimulation platforms like electrical simulations and what other kind of feature sets that we're talking about, and how we're going to manage the power and building the system inside. So yeah, so that will be my answer for scaling up.
I think for small animal, it's actually harder than do it in a larger animal, because in small animals the space is quite limited, so we need to make everything miniaturized. But for a larger animal, I think we can make the receiving antenna larger, so we get more power, and we will have more electronics to control it, and we can probably put a battery like there to like increase the power. So I would say it's going to be easier to do it in larger animals than small animals. Doing it in mice is like really hard.
Yeah, I think Einar wanted to ask again. Did it work to bring him up? Einar, and I invited you to speak in case you wanted to ask another question. And yeah, it's not working. Okay. In the meantime, what is the operating frequency of the power transfer? Asked Benjamin in the chat.
Operating frequency meaning the RF frequency, or if we're talking about RF frequency, it's 13.56 megahertz. If that's what you're asking for, is it? Or are you talking about the operation frequency in terms of the frequency of like either LED? Okay. Oh yeah. Okay. Yeah, I saw the RF frequency. Okay. Nice.
Yeah, great. I don't. It doesn't work to bring Einar up. Does anyone have last questions? I'm not sure how much time you still have, Yiqing and Mingzheng, to answer questions, because we already took more than an hour from your life to do this. So
Yeah, it's so much fun.
it is fun. Oh, great! Yeah, I'm glad. I'm always happy to hear that when the guest speakers also have fun and it's not just parasitic relationship. Um, um, I saw that your micro LEDs. So you're planning on switching to deep red light, right? Like near infrared light. Is that right? So you have contracts. Did you get contracts like optogenetic contracts? Maybe from Peter Hageman. I think
I
you
think
were.
I use crimson for red shifted crimson R.
So in theory, you could have both. Then, like you could have an. Could you have an inhibitory and excitatory and different wavelengths? Like, do you think that would make sense for your experiment setups? Because then you could have, if it's micro LEDs, you could have like strings of different lights, maybe.
Yeah, sure. We can use blue light and red light. And I would say I slightly prefer to not use inhibitor option. I'll just, like for that purpose, I will express excitatory option in inhibitory neurons instead for inhibition. So that's my personal preference, I would say.
Yeah, that's interesting. I think to go that route, because I think so many also disorders are very GABAergic driven. And are you planning on maybe try out glia cells like astrocyte expression and modify? Because there's more and more research now pointing towards that the glia cells like oligodendrocytes, astrocytes in specific, play a huge role in different type of disease states, including neurodegeneration, ALS, and so on. So it would be really interesting to look at that too. Thank you.
Yeah, yeah, it's definitely doable in clear cells. And we can, like with the fluidic system, we can express these like chemogenetic receptors and use like for example CNO to change their like intracellular signal pathways, for example.
Yeah, that's really exciting because, if we do it pharmacologically, the traditional way, we kind of, I think most people inhibit gap junctions, but it's all over the brain and it can be really harmful and not have, generate inconclusive results. So to be more, to achieve precision with or glia cells, I think would be really interesting. And welcome Abyss to the stage. Did you have a question?
Hey Kat, hi everyone. Yeah, I guess I have a question, although like I joined the discussion much later. It's based on the question that you actually raised, which is, um, what is the sort of like the advantage of using blue light? First, I would like to commend you that this is a very astonishing work. But and that being said, my question would be, um, instead of using the blue light, which I think is kind of typical for opto experiments, since you're using wireless power transmission, why not focus on the red or near infrared spectrum to essentially activate your channel Dobsons? Because one advantage that I can see is that first, the power required to actually induce red light would be far more smaller, and um, essentially you can access also much deeper level of tissue by using red and infrared. So I'm just wondering, that will be my question or comment, if you will.
Yeah, I totally agree that red light is a better option, and that's what we are trying to use right now. I think from now on we do prefer to use red light and red shifted options than like using like channel Dobson.
Gotcha. Thank you.
Yeah, thank you so much. If anyone has, let me check the chat again. Well, oh, I see Benjamin. You have a question. Let me try to invite you to speak. And Einar, I still couldn't bring you up. Benjamin at work, but Einar, it's not working. Hi, Benjamin. Maybe the last couple of
Hi,
questions.
I
Go.
enjoyed the article and I asked the question in the chat and I got an answer, so thank you.
Okay, great. Einar, I'm not sure why it's not working to bring you up to speak. Maybe. Oh, he wrote in the chat. Are the mice being set up with the monitors as time let the mice the 2 week to have a monitor? Um. So I guess is it the question, Einar, to how the mice are being monitored in the meantime while the virus while the expression is going on in the brain of the virus? Is that what you mean in those 2 weeks?
Yeah, exactly. Is there a time difference in where the the mice here are actually being adapted to be um, focused on with there's certain devices going on and optic monitors. Uh, is there a time span here with there some conscious related animal um, being fully set up into an experiment, and some are partially being set up uh, because they adapt as all conscious beings. Yeah, thank you.
Yeah, I will let them recover for that virus for virus expression. I will give them some time to allow the virus to express. That's typically more than 2 weeks. So at that point, the animal are like adapted to the devices and like recovered from the surgery. Uh, yeah, although we can do it like for farm a photopharmacology experiment, we can do it faster, like maybe 3 days after the surgery. But I would prefer to let the animal to recover a little bit more from the surgery and get used to the device before I'm doing like behavior experiment. Although we can do it like as soon as 3 days.
That's very interesting. That's very interesting. Do you see a difference in behavior after the experiment has been done?
We typically don't monitor like behaviors after the experiment because we need to sacrifice the animal for histology and other validations. But I think for most of animals that we accidentally leave on, so they do not behave much different than their like baseline level. But I've never monitored it like a long time after the experiment because it's not like allowed by iCook. So
Well, it's an interesting question, Einar, and there are labs that are interested in the ultra sonic communication between mice and also rats. And for example, a friend of mine, Marie Monfils, she's at Texas Austin University. She does a lot of research like that because she's looking into fear memories and extinction memories and so on. And she did some experiments where they went through the fear memory, Pavlovian fear conditioning, the rats, and then she checked if they warned the other rats in the colony of some of some threat coming, and she could then see that the following rats, usually you separate them and soundproof and so on, but she did the opposite for those experiments, and she found that they actually warn each other, and the following rats that were kind of warned, they they would learn the fear conditioning faster. And yeah, there's a lot of intercommunication with ultrasonic sound recordings, and so that go, but they mostly focus on fear conditioning, which is quite stressful event. But it's an interesting question. It's actually pretty straightforward nowadays to do that. They are like recording devices, and there are specific trained neural nets to kind of analyze the data, and it's pretty easy and straightforward to do it if people are interested in. So yeah, if you're interested in look at papers with like ultra, with their that look into these vocalizations of mice and rats, and you should find something there. Thank you.
Thank you very much. Thank you.
Abyss, you had the last question, or did you want to comment? I saw you on mic.
Yeah, it's probably like a last question. Um, so I noticed that you actually kind of offboard the signal processing to a computer through the bidirectional data transfer that you have. Is there any chance that you're looking into doing on-chip kind of data processing? And if so, if there are, if you're actually considering um, algorithms or basically like programs that can essentially utilize low power, given that your source would be radio frequency at this point. Just curious.
yeah, I think so. The on-chip processing is definitely or will be like faster. And it's definitely like something we're currently working on. I think for the closed loop system we're talking about, so it's still not the on-chip processing, but I would say like it depends on the sensing modality, like what is the sensing modality we're using. Because if it's electrical sensing for in vivo phase experiment, it requires a high like temporal resolution. So I think for that purpose, like the on-chip processing is definitely better than like doing it like with a bidirectional communication. But I would say for a relatively like other neural signal, for example EEG and maybe calcium transient, so the temporal resolution requirement is not that high. Probably doing it with like the computer would be easier to do.
Yeah, I would say for device perspective, for now the on-chip processing is going to consume a, 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 a large amount of power at this moment. Even though we use NFC, we if we want to do micropump simulations plus like the micropump delivery, the optical simulations at the same time, plus the on-chip processing, we probably definitely need like further developments into how to manage the budget, like the power budget at this moment. But yeah, in the future, for sure, this is something that we haven't integrated in our system, but in the future, for sure, yeah, it's possible.
Thank you. Mike, over to you, Kat.
Well, thank you so much for presenting this amazing work and technology and explaining it so well and answering so many questions of ours. I hope maybe you'll come back one day when maybe you have update or something new that you can share with us. That would be really wonderful. This was a great honor to have you here, both of you, and we wish you all the luck, although I don't think you need it, but all the luck and funding to continue doing the work that you wish to do and out of curiosity and doing good in the world. It's always beautiful to see people that they use their brain for the good of humanity. So thank you for doing that, and yeah, we wish you all the best and happy weekends. Thank you.
Thank you, everyone.
Thank
Thank you
you.
so much.
Okay, yeah, have a great weekend, everyone, and we'll close the room in 3, 2, 1. Bye, everyone. Thank you.